afternoon. I'm 승엽현 from Seoul National University, and I'm very honored to receive 이세영 Outstanding Award and have a chance to introduce my work from PhD course. Lung cancer is a commonly diagnosed cancer and is the leading cause of cancer-related deaths in worldwide. Malignant tumors are composed of heterogeneous population, including CSC, which is cancer stem cell. CSC is a rare population of tumor that carrying cancer stem cell-like property. CSC can generate tumor via self-renewal and differentiation into other subtype of tumor cells. CSC also causes tumor relapse and resistance. Therefore, understanding the underlying mechanism of CSC is essential. However, due to the heterogeneity of CSC and lack of specific and reliable marker for CSC, current anti-CSC therapy is limited. So, I tried to identify novel marker and novel strategy targeting CSC with already known their properties. I'm going to talk about my results in two parts. Firstly, I would like to introduce about ninjurin-1 as a novel marker for CSC. Ninjurin-1 is a well-known cell adhesion molecule. However, their functional role in cancer is still controversial. So, to assess the clinical relevance of ninjurin-1 in lung cancer, ninjurin expression was examined in human tissue and public database. Ninjurin-1 was significantly upregulated in tumor tissue, and NSCLC patients with high ninjurin expression show poor survival ratio. To assess the functional role of ninjurin in lung tumor development and progression, mice with lung-specific ninjurin induction were utilized. Ninjurin-1 was induced in CCSP-positive club cell and SPC-positive alveolar type 2 cell. Ninjurin mice show significantly increased lung tumor formation. In addition, ninjurin mice show obviously worse overall survival compared to their control mice. To investigate the role of ninjurin-1 in CSC, ninjurin expression in potential CSC-like subpopulation was assessed by using their general properties. Ninjurin expression was increased in potential CSC-like population. To provide direct evidence for the functional role of ninjurin-1 in the acquisition of CSC properties, ninjurin-modulated stable cells were established. Importantly, the functional role of CSC were regulated by ninjurin-1. Consistently, the in vivo limiting dilution assay showed that ninjurin-1 high cells show greatly tumorigenicity than compared to their corresponding ninjurin low cell. To validate the clinical relevance of ninjurin-1, ninjurin-1 expression was analyzed in potential CSC-like subpopulation within patient-derived tumor. Ninjurin expression were also increased in CSC-like subpopulation of patient-derived tumor. Furthermore, ninjurin high subpopulation in the patient-derived tumor show significantly increased CSC property compared to their corresponding ninjurin low subpopulation. Since Wnt, Petio, Notch, Hippo pathway are well known as a critical regulator pathway in stem cell function, targeting of this pathway were analyzed to discover how ninjurin regulates cancer stem cell function. Only targeting of Wnt beta-catenin pathway were regulated by ninjurin-1. In addition, beta-catenin, which is the key regulator of Wnt canonical pathway, was also regulated by ninjurin-1. However, there were no significant change in Wnt non-canonical pathway components. So, Wnt beta-catenin pathway activity were regulated by ninjurin-1. To investigate the mechanism by which ninjurin activates the Wnt signaling pathway, entire mRNA expression of Wnt signaling components were validated. Most Wnt signaling components were either decreased or remained unchanged in ninjulin cell. Therefore, the impact of ninjulin-1 on the Wnt signalosome complex formation was focused. Immunoprecipitation assay showed that ninjulin-1 associated with Wnt signalosome complex, and pull-down assay confirmed that N-terminal domain of ninjulin-1 participates in this association. So collectively, this result suggests that N-terminal domain of ninjulin-1 make a complex formation with Wnt signalosome and activate the canonical Wnt beta-catenin signaling pathway. In summary, ninjulin-1 is a potential CSC marker of NSCLC, and ninjulin targeting therapy can be combined with other anti-cancer drugs for cancer treatment. As a second part, I would like to introduce Fischer protein system as a potential target for eliminating CSC. The Fischer protein system is mainly composed of Fischer protein 70 and 90. Main function of Fischer protein system is controlling the proper folding and stability of client proteins. And Fischer protein system maintains cellular homeostasis against many environmental stress. Fischer protein system are frequently deregulated in various of tumor, and Fischer protein system also known to be closely associated with CSC function. Therefore, targeting the Fischer protein system could be an effective strategy for eliminating both CSC and non-CSC population. Based on previous study that suggests the impact of Fischer protein system on CSC behavior, expression profile of Fischer protein system components were assessed in CSC-like subpopulation. Then, the gene expression of Fischer protein system mostly increased in CSC-like subpopulation, and especially HSP70 gene was mostly increased. To discover hit candidates that could eradicate both CSC and non-CSC population by targeting the Fischer protein system, a natural product library was screened by using OCT4-GFP reporter cell. Then ebodiamine was identified. Ebodiamine displays potent anti-CSC activity in a concentration-dependent manner. In addition, ebodiamine shows anti-tumoral and anti-CSC effects in KRAS spontaneous mouse model. To determine whether Fischer protein 17 is a cellular target for the activity of ebodiamine, a series of experiments were performed. The DART analysis, biotin avidin pull-down assay, ATP agarose pull-down assay. This assay showed that ebodiamine binds to Fischer protein 70 N-terminal domain, especially via ATP binding pocket. Then this binding induced the protein degradation of Fischer protein 70 via ubiquitin-mediated proteasomal pathway. The binding mode of ebodiamine to Fischer protein 17 was confirmed by docking analysis. In addition, point mutation at ATP binding residue almost completely diminished the interaction between ebodiamine and Fischer protein 70. These results collectively indicate that ebodiamine disrupts the Fischer protein system by directly binding to the N-terminal binding domain of Fischer protein 70 and causing its destabilization. I can conclude that Fischer protein 70 is a potential target for eliminating CSC, and ebodiamine acts as an effective Fischer protein 70 targeting anti-cancer drug, eradicating both CSC and non-CSC population. To close, I would like to express gratitude to my professor, Ho-Young Lee, who gave me a lot of advice and guide my research. And also, I thank to other professors, many collaborators, and my lab members. Thank you for listening to my presentation.